Oof. Right. So, see, we were talking about. So, there's a lot of problem, especially in the last half an hour in the last lecture. So, let me recall. We were talking about blood, right? We were talking about the blood, and I told you blood is it's a fluid, connective tissue, and the pH of blood is 7.4, right? And this blood is formed by bone marrow, right? And average volume of blood is 5 to 5.5 liter, right? And the blood is river of life because it helps in the transportation of what? All water, oxygen, food, hormone, etc. Fine, right? Fine. So we are just talking about we are just talking about chemistry of the blood. Hmm? This is chemistry of the blood we are talking about. So even I told you this blood is fluid connective tissue. Hmm? And this blood has two main components. One is plasma and second is corpuscles. One is plasma and the second is corpuscles. Right? So see, when I come to this blood, when I come to this blood, whole blood, this blood has two components. One is plasma and second is formed element. This formed element is corpuscles. This plasma is 55 to 60%. This formed element is 40 to 45%. This plasma has so many components like this. First is the amino acid, second is nutrients, third is protein, this fourth is the nitrogenous waste, fifth is electrolytes, sixth gases, and this protein has so many. This protein has so many components like albumin protein, adenosine protein, globulin protein, and even antibody. Antibody. Fine. Antibody. Fine. This antibody. So nowadays you heard about plasma. Plasma therapy. You heard about plasma therapy. Hmm? So this plasma of that make a sense of the that make a sense of the plasma of COVID survivor has sufficient amount of antibodies. Sufficient amount of antibodies. The plasma of COVID survivor has sufficient amount of antibodies that we are extracting from the body of corona survivor and then injecting, then extract separating this antibody from the plasma of COVID survivor and then transfusing this antibody to plasma to COVID patient as a therapy. That is the plasma therapy nowadays we are talking about. Fine. So when we talk about the plasma therapy, it means as a result of the corona survivor, as a result of corona survivor, that survivor has sufficient amount of the antibody which is quite effective against corona, which is quite effective against corona disease, and this antibody we need to extract from the body of the corona survivor, and we are not supposed to take this antibody separately, rather we are extracting plasma from the body of the corona survivor and then transfusing it to the body of corona patient just to make his or her just to make him healthy as a medication. So that is what it's a plasma therapy, right? So that is the one aspect of this plasma, that is the component of the blood, and the second aspect of this blood is it's a formed element, and the formed element, it has three components. One is the leukocyte. Leukocyte is the WBC, and the WBC has five. And the function of WBC is what? The function of WBC is immunity. The function of WBC is immunity, and this WBC has five different types of WBC. One is neutrophils, second is basophil, third is eosinophil. Yes, this is the one, two, three, four is lymphocyte, and fifth is monocytes. Neutrophils, basophils, eosinophils, lymphocytes, and monocytes. Five different types of leukocytes or WBC. Five different types of leukocytes or WBC involved in immunity, and we have discussed about cellular immunity. We have discussed about. Are you getting my instruction properly? Are you getting my instruction properly? Are you getting my instruction? Are you getting my instruction properly? Right. Fine. Second is the platelets. Okay. Platelets. Platelets means thrombocytes. 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 And this platelet or thrombocytes helps in blood clotting. It helps in blood clotting. Okay, fine. While this erythrocyte is RBC, erythrocyte is RBC. I'm coming to this. I'm coming to Namrata. I'm just coming to your question. Well, the RBC is erythrocyte, and the RBC has hemoglobin. RBC has hemoglobin. RBC has hemoglobin, and this hemoglobin helps in oxygen transportation. This hemoglobin helps in oxygen transportation, right? So from here, what we need to conclude from this entire discussion, one is what is plasma? Plasma is component of blood. Component of blood. Blood is not a single unit. Rather, blood is a complete unit in the form of the tissue, and this blood has so many different components. One component is plasma, and the plasma is placed for so many components, and here antibody protein is one of the component of the plasma which is developed against infection. So now this question was asked by someone else like Namrata. This question was asked by this question was asked by Ashish. Plasma therapy is also applicable to many different curable diseases. Yes, yes. Plasma therapy. This could be used against any kind of infection, because infection means always pathogen. Against any pathogenic infection, your body responds, and this response is in the term of antibodies, right? And the place of antibody is where exactly in plasma. Fine. So nowadays we are talking about this plasma therapy because in the case of the coronavirus, in the case of the COVID, we don't have other medication. We don't have other medication. That's why we are just talking about the plasma therapy nowadays, right? Otherwise, in most of the cases, means in the different examples of the different categories of the disease, we do have the other provision of the medication. That's why we are not talking about the plasma therapy in other cases. But even if you want to apply this plasma therapy in any pathogenic infection, we can apply, because against any pathogen, body respond in the term of antibodies. So right, so we need to extract that plasma, clear, and we need to extract antibody from the plasma, and that antibody is pathogen specific antibody that we need to transfuse to the body of that patient, right? So we can use anywhere. In the case of pathogenic infection, we are just talking about the pathogenic, but in most of the cases, in most of the example of the pathogenic infection, we also have the main stream of medication. That's why we are not talking about the plasma therapy. And in the case of coronavirus, we don't have medication, right? We don't have mainstream medication. That's why we are talking about plasma therapy. But still, plasma therapy is not 100% authentic and reliable. Means we cannot apply this plasma therapy as a thumb rule. That's why it is not been recognized. Government is demanding, government is announcing, right? Everyone whosoever has recovered from corona, uh, uh, come forward and donate plasma. But even we cannot, even it is not confirmed by WHO. We could apply this plasma therapy as a thumb rule, and this is going to be pure this corona patient 100%. Clear? So normally it is applicable. Normally it is applicable. It is effective in most of the cases, but it is not effective 100% in all cases. It is not effective 100% in all cases. And even every corona survivor is not going to have that much of antibody which is required for the diffuser, right? Because the level of antibody is variable from person to person. So probably you have recovered against the corona, but even unless your body is going to respond that much of antibody, that is the threshold value, means ultimately body has to maintain the threshold value of the antibody, right? And after maintaining this threshold value of antibody, you will be acting like plasma donor. So everyone is not supposed to act like plasma donor before plasma donation. If you have corona, if you have recovered against corona and you are willing to donate your plasma, so before taking your plasma, they are going to check your antibody value. And if your body is responding antibody to that extent, in that case, they are going to take your antibodies through the plasma. Otherwise, they will refuse. Fine. So that is the reason behind we are not using this plasma therapy as a universal concept, but even it is the most effective concept, right? The first thing is what. Again, the second thing I told you in the last lecture, means that last lecture was quite disturbed. The last 1 hour was quite disturbed. But in the earlier, I told you, blood is named after antigen, right? Blood is named after antigen. Means blood A is A because there is antigen A. Blood B is B because there is an antigen B. Blood AB is AB because of antigen A and B. Clear? And the blood O is O because of the antigen O. Right? And the location of antigen is always, the location of antigen is always, the location of antigen is always, the location of antigen is always at the surface of RBC. Even I told you, at the surface of RBC. The location of antigen is always at the surface of RBC like this. This is A or B or both AB or no. So when we talk about the blood group, it means the blood group is completely depending upon. The blood group is completely depending upon the erythrocyte, and the erythrocyte is the part of formed element or the corpuscles. Means this blood group is completely depending upon the formed element or the corpuscles, while blood group is not associated with the plasma. So when we talk about the plasma, and nowadays we are simply misleading, like the B positive blood group plasma is required. That we are not supposed to do like this. Why? Because plasma is always neutral. And any blood is going to donate blood for the plasma. In that case, they are going to extract plasma from the blood group of any specific blood group, which is the A positive, A negative, B positive, B negative, AB positive, AB negative, right? O positive, O negative. So the plasma is without blood group, right? The blood group is only the part of the formed element, So this is simply, it's a misleading, like the B positive plasma is required. It does not make a sense. Because when we talk about the A positive, B positive, AB positive, O positive, A negative, B negative, AB negative, O negative, it means we are talking about simply the formed elements or the corpuscles, especially RBC. And it is not related with the plasma, though both are the part of the blood. So the plasma is different and the formed elements are different, and the blood group is positive or negative. Blood group is the AB and O. That is the part of the corpuscles. Right? So plasma therapy is independent and the neutral of the blood group. The same blood group plasma is required to transfuse, just to avoid complications, just to avoid complication, because apart from what we have discussed so far, blood has so many other components. Blood has so many other components because it's a group of cells. It's a group of different type of cells, and we have discussed only the important types of the cells, right? So normally we are normally we are we are using the same blood to plasma just to avoid the other complications. Otherwise, right, even the other group plasma could also be used for the medication of the corona patient. I hope it is clear to everyone. So this question was asked by Namata. This question was asked by Ashish, right? This question was also asked by Nayana. Nayana, I hope you got your answer. Madhu, will all question come from the current affair from the life science? Huh? This Madhu mentioned this statement so many times. Madhu, it's not life science. We have mentioned this heading as a life science. But we are talking about the 50% of the components of the science and technology. So here this topic is the life science. But here we have included so many aspects. We are going to discuss, we are discussing about the biotechnology, we are discussing about the health issues, we are discussing about the disease aspect, we are discussing about the physiological. So please, you just try to see the first lecture. We have already demarcated what actually we are going to continue in the class. So officially this heading is the life science, but unofficially we are going to discuss more than 50% of the topic which is required under the head science and technology. So please be specific, and then you try to make a statement, please. So can we go ahead? I I hope you got your answers. Siddhant, what is the structure of antibodies? See, Siddhant, that much of discussion is not required here. Why you are talking about? No, I'm just telling you. I'm just spending 30 seconds. The structure of the antibody is variable. Structure of antibody is variable, and if I talk about this antibody, I'm just giving you the one example of the antibody. See, this is see here. It's a immunoglobulin Ig antibody. It's an immunoglobulin, that is Ig antibody. It's quite complicated, and when you see here, this consists of, it's a chain. It's a long chain of amino acid. Clear? This is called as peptide chain. Clear? So if you just try to count here, see here, 1, 2, too long peptide chain, right? Too long peptide chain, and see, 3, 4. It's a short peptide chain. 3 and 4. That is short peptide chain. So it's a quite complicated structure. It's a quite complicated structure, and even like this, it looks like this, and it consists of a number of receptor sites, and these receptor sites are going to attach pathogenic infections, and it does not allow the movement of pathogen inside. That is going to create a defense mechanism. So it is acting like protecting shield, because this antibodies, the surface of antibody has so many reception site, and the purpose of this reception site is what? Just to welcome this pathogen, right? And the welcome to the pathogen is required to neutralize the action of this pathogen. And once this pathogen is trying to intrude this antibody receptor site like this, right? If it is trying to intrude the antibody reception site like this, right, it is going to infect you. Otherwise, as long as this antibody is going to prevent the infection, intrusion of this pathogen infection inside, it will protect you. That's why we have discussed in the first lecture why this antibody is called as the protecting layer of the body, because it is going to protect you against pathogen infections, right? So there's, right, we have so many antibodies, and the structure of antibodies are variable because these are made up of amino acid peptide, and the peptides are made up of amino acids. So in few antibodies, the number of antibody and the number of amino acids are maybe 50, right? In some case it's 100, in some case it's a 150, in some case it's a 500, right? So as per the involvement of the amino acids, the structure of antibodies are variable. So we don't have common structure which is applicable to all antibodies. The common discussion is what antibodies are peptide, means protein in nature, and made up of amino acids, and because of the differences in number of amino acids, the structure of antibodies are variable. Fine? Okay. So this is all about the discussion, what we have done so far. Now, listen, after doing this, I am coming to the, everyone is getting this video properly, right? If someone is not able to get this video properly, because majority of the students, they are responding like the audio and the visual reception is proper. From my side, it is showing that, the reception is proper, right? So if someone is not able to get this probably because of local area network. Yesterday it was a problem because we experienced the heavy rainfall in Delhi, and especially in my area. That's why video was just taken to everyone. But now this time, everyone is getting a proper video, audio, and the visual reception except the few. So that is because of your local area network. Now coming to the next part of the discussion, see what we have discussed. This chromosome, this chromosome is gene carrier, fine. And when we talk about this chromosome and the gene, both are made up of nucleic acid, and the nucleic acid is nothing but it's a DNA, RNA. So this part is quite important to everyone, because this time we are going to discuss about structure, function, gene, defect, and gene therapy. Clear? We are going to discuss about the structure of DNA and RNA, function of DNA and RNA, genetic defect, and gene therapy. The entire topics we are going to discuss in one shot in today's lecture. So, Kika, if a person blood group is AB positive, right, and is infected, will he need more care since antibody is not there in the blood group AB positive? See, Tokika, when we talk about the antibodies, we have n number of more than 100 types of antibodies are there in the body. More than 100 types of antibodies are there in the body, right? And in the blood group AB, both antigens are there and the antibody is absent. It means what we are trying to make this statement here. Now the antibody is absent in the term of blood group. right? In the blood component, antibody is absent. So in the blood component, antibody is absent. Otherwise, there are so many other categories of antibodies have already been there in the body of the blood group, right? Man with the blood group AB. So it is not like that if anyone is having AB blood group does not have antibody in the blood group, it means he or she is not having other examples of the antibodies, right? He or she must have other categories of antibodies, but antibody in correspond to the AB blood group is absent, right? And even it is natural, so there must be the natural compensation of the antibody in the other aspect. So it is not like that the AB man doesn't have any antibody. If it is like that, that man is not going to be survived. So that man has, right? Suppose if someone has 100 antibodies, right? If suppose if I have 100 antibodies, so and I belong to blood group O, fine. So I have one of the antibody in the blood group. One of the antibody is what, it's a AB that I have, and if it means the 99 other antibodies apart from the blood group A, apart from the blood antibody A and B, I also have the 99 other categories of the antibodies. But in the case of man with blood group AB, right, if that man does not have antibody especially related to the blood, so it means that man has 99 other antibodies. And not only 99 other antibodies, definitely that man has one additional antibody, because in blood antibody is absent. So in that case, naturally this man is evolved. This man is evolved naturally because blood group is natural. So this man is evolved naturally, and if antibody in AB blood is absent, so that absent antibody must be compensated somewhere. Otherwise, what happened? If that man is not going to have that much of antibody, so definitely what happened? This man will feel more pathogenic. And more pathogenic means what? This man is vulnerable to the pathogen infection. And if this process is going to be continuous, so definitely there could be a chance of extinction of this man. And it is not like that. So that it means it's a natural adjustment in the process of evolution. It's a natural adjustment. So the absent antibody in the blood must be compensated somewhere in the body in the form of other antibodies. So coming to this nucleic acid as a DNA and RNA, coming to this nucleic acid as a DNA and RNA, see, chemical structure of gene is nucleic acid, and this nucleic acid is DNA, RNA. This nucleic acid has three components. One is base, second is sugar, third is phosphate. Yes, this base is nitrogenous, sugar is 5 carbon. 5 carbon sugar means pentose sugar, while the phosphate is H3PO4. H3PO4, right? So here in the among the nitrogenous bases, there are two different bases. Like one is furan, and the second is pyrimidine. One is purine and the second is thymidine. Fine. This purine has two classical examples. One is adenine, second is guanine, adenine A, guanine G. This pyrimidine has three classical examples: uracil, cytosine, and thymine. Fine. So total five bases. Total five bases. This purine, two bases, adenine, guanine, uracil three bases, pyrimidine three bases, uracil, cytosine, thymine. It's a U C P. Total five bases, nitrogen bases. Okay. Sugar, pentose sugar. One is, there are two pentose sugars. One is ribose and the second is deoxyribose. One is ribose and the second is deoxyribose. One is ribose and the second is deoxyribose, RIBOSE. And deoxyribose means DEOXYRIBOSE. Deoxyribose and oxyribose. And this H3PO4, that is very common in both DNA and RNA. Fine. Right? So listen, here, if we talk about difference between this DNA and RNA. DNA has adenine, guanine, thymine, cytosine. RNA has adenine, guanine, uracil and cytosine for the bases. Means there is a one point of difference, and this is what? This thymine is replaced by uracil. The difference between DNA and RNA. Sugar, DNA has deoxyribose deoxyribose sugar, and this has ribose sugar. While in both of the cases, we do have a provision of same as a H3PO4, H3PO4, and common to both of the cases, H3PO4. Right? Fine? Okay? So listen, look here, look here. This is the double helical structure of DNA. Hmm? And this DNA has, see here, DNA has a number of bases like pyrimidine, cytosine, thymine, and one more is here. It's a uracil. Structure is not required. Structure is not required. This purine has two examples like guanine and adenine. Structure is not required. Clear? So total 1, 2, 3, 4, 5. Total five types of bases are there what we have mentioned so far. And out of here, sugar, deoxyribose and ribose. Sugar, ribose and deoxyribose. Ribose and deoxyribose. Sugar. And there's one point of difference. 5 carbon sugar. You see here. It's a 1, 2, 3, 4, 5. 5 carbon sugar in both of the cases. And the one point difference would be in the ribose, there's a hydroxyl group which is replaced by deoxyribose, where's the hydrogen. So the only difference between the deoxyribose and the ribose is what? The second position is occupied by hydroxyl line in the case of ribose, which is replaced by the hydrogen in the case of deoxyribose. That is the simple difference between deoxyribose and ribose. One point of difference between deoxyribose and ribose. Clear? And this ribose is in the case of RNA, while deoxyribose is in the case of DNA. Sugar molecule as a sugar, as a sugar. Fine? Okay? Right? And this third is the H3PO4 phosphate. This third is H3PO4. It's a phosphoric acid. It's a phosphoric acid. Fine? Right? And see here, See here, this one sugar, one base, and one phosphate. This is one nucleotide, one nucleotide, one base, one sugar, and one phosphate, one nucleotide. Clear? And again, just make one more nucleotide like this, one more nucleotide like this, like sugar, base, phosphate, sugar, base, phosphate, nucleotide number 2, nucleotide number X. So here what is going to be done? See, in all cases here, the polymerization between the first and the second nucleotide, the second and the third and the fourth. So this polymerization is going to be done. This is polymer bond. And as a whole, the entire structure is called as what? Nucleic acid. Right? Nucleic acid. And this nucleic acid is nothing but it's a polymer of nucleotide. And this nucleic acid is, if it is double stranded, so this is DNA. And if it is single stranded, this is RNA. Single stranded, this is RNA. Okay. So this double stranded DNA, this double stranded DNA, double stranded DNA is because of base pairing like AT and GC. Adenine pairs with thymine and guanine pairs with cytosine. So DNA is double stranded because of the base pairing, while RNA is single stranded because there is no base pair. There is no base pair. There is no base pair. Right? There is no base pair. Is that clear? Is it clear? Fine. So listen, if coming back to this structure, like DNA, you see here, if I am going to elaborate this DNA like this, it's a strand number 1 and the strand number 2. Let it uncoil. let it uncoil. Like this is the one strand of DNA, like this, and this is the second strand of DNA, like this, like this. Fine. This is the one strand here, and here is the strand number second. Fine, and we have mentioned in uncoiled form, and here nucleotide, 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 nucleotide. It's a series like this. Fine, and here there's a sugar phosphate, and there's a base like A. Sugar phosphate base like C. So in the second strand here sugar phosphate, sugar phosphate. Here must be base like T. Here must be base like G. So in DNA, base pairing is always specific, and this specific base pairing is what it's a AT and the GC base pairing. So this is what it's a base pairing, and this is AT and GC base pairing. This is quite specific in DNA. Fine, it is quite specific in DNA, and you see here, if you see here, see here, like this. Look here, it's a, see, this is the base pairing number one, base pairing number 2, base pairing number 3, 4, 5, 6, 7, 8, 9, 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So in DNA, this is what? It's a one coil of DNA, which is 34 angstrom. This consists of 10 base pair. The one coil of DNA consists of 10 base pairs. One coil of DNA. How the length of DNA is measured? It is by measuring coiling of DNA, and the one coil of DNA consists of 10 base pair, and its length is 34 angstrom. Fine. So this base pairing is the characteristics of only DNA, and it is not in RNA. That's why RNA is single stranded. And if you just try to see the lecture number 1, fine. See the lecture number 1. We have discussed about the structure of the coronavirus, that is the SARS-CoV-2, and I have shown you the single length of the virus, the single length of the RNA in the case of the coronavirus. So in all cases of mainly we can see the RNA as genetic material mainly in viruses, and in all categories of the viruses, RNA is usually single stranded in nature, just because of AT and the GC base pairing is not applicable in the case of the RNA. And it is not like that this thymine is replaced by uracil in the case of RNA. So this A must be paired with the U. Not at all. It is not there in the case of the RNA, right? So this AT and the GC base pairing is always specific in the DNA. And it is not only that, this also counts the functionality of the gene. So the gene is normal and functional. When we talk about, just mark my word, just mark my statement, this statement is going to be realistic and it is going to be coded for you. Why? Because when we talk about the gene is functional and the normal. So when we talk about the gene is functional and normal, it means there must be AT and the GC base pairing, right? So as long as base pairings are normal, it means gene is quite functional and the gene is quite normal. So for the functionality of the gene, for the normalcy of the gene, there must be the AT and the GC base pair. Why? Because I will let you know why. Because all genetic informations are hidden in bases. Right? I am tall because I inherited the gene from my parents, and the gene consists of three components. And these three components are what? It's a sugar, base, and the phosphate. And the genetic informations genetic informations are hidden only in bases, not in sugar and the phosphate. So sugar and the phosphate are structured in nature, while the genetic informations are hidden only in bases like A, B, G, and C. Clear? So just mentioning here in the box I'm mentioning here, genetic information are lying with ATG and C, means bases, while while sugar and phosphate and both are structure in nature, both are structured in nature. Fine, structure in nature. And the brief introduction about the DNA is already mentioned in your booklet. It is already mentioned in your booklet, right? So if you, because this is the static part we are discussing. This is the static part we are discussing, and then we are going to make it dynamic as per the requirement of the exam. So as long as if you want to discuss about this static part, this has already been there in your booklet. But these, this particular discussion is required just to make it more dynamic, and from where UPSC is going to get question these days, right? So this is what here. So if you allow me to show here, see, it's a strand. Look here. This is the structure of DNA. This is what it's a DNA. It's a DNA. This is the one strand of DNA. It's the one strand of the DNA. This is the second strand of DNA. It's a base like A is there, pairs with T. This G pairs with C. This T pairs with A. This C pairs with G. Always between A and T. Between A and T, there are two bonds, and between G and C, there are three bonds. And these bonds are 3, 2, and these bonds are hydrogen bonds. These bonds are hydrogen bonds. These bonds are, so the AT base pair has 2 hydrogen bonds, while the GC base pair has 3 hydrogen bonds.